Hello and welcome to this GCSE Science revision video for additional biology on enzymes. In this video we'll be covering the function of enzymes, the factors that affect enzymes, enzymes in digestion and the uses of enzymes. Enzymes are biological catalysts which speed up the rate of reaction within living organisms. Organisms or the cells within organisms would often not be able to function without the use of enzymes, as the reactions that was happening within them would happen too slowly. There are many different types of enzymes, however they are all proteins that have been folded into a complex shape. Enzymes are specific and only allow one substrate or reactant molecule to fit into them at one time. The area that these substrates or reactants fit into is called the active site. We start off with a reactant or a series of reactants and an enzyme. Those reactants fit into the active site and form either a reactant enzyme complex or reactant substrate complex. That reactant is then broken down into two separate ones, which are then released. Because an enzyme is a catalyst, it can then go on and be reused to catalyze other reactions within the cell. If the shape of the enzyme changes, its active site may no longer work. We say that the enzyme has become denatured. Enzymes can be denatured through high temperatures, which break up the structure of the protein, or extremes of pH that cause the charged areas within the active site to no longer work. It's worth noting that we can't say that the enzyme has been killed, because enzymes are only protein, so are not alive. There are a few factors which affect enzymes. Temperature has a great effect on enzymes. The graph shows that as the temperature goes up towards 40 degrees, the rate of reaction increases. This is because particles will be moving faster, so the enzymes will be moving quicker, so we'll be able to carry out more reactions. However, after 40 degrees, the rate of reaction goes down drastically because the enzyme will become denatured. If the enzyme becomes denatured, it will no longer be able to carry out its function, as the substrates will no longer be able to fit into its active site. It's worth remembering that body temperature is around 37.5 degrees, which is the optimum temperature for enzymes to work at. Changes in pH also alter the enzyme shape. Different enzymes work better at different pHs. So, for instance, an enzyme that works best in your mouth will work best at pH 7. However, an enzyme that works in your stomach will work best at around pH 2. The graph shows how an enzyme has an optimum pH. Different enzymes will have optimum pHs that are different to each other, some being higher, some being lower. There are many enzymes involved in the human body. Many different types of enzymes are used in digestion to ensure the breakdown of food so that it can be absorbed. The enzyme amylase breaks down starch into sugars. The enzyme protease breaks down proteins into amino acids, and the enzyme lipase breaks down lipids into fats and glycerol. Amylase is produced by the salivary glands in the mouth, the pancreas and the small intestines. Protease is produced in the stomach, by the pancreas and in the small intestines, and lipase is produced by the pancreas, and you find it in the small intestines. So in summary, amylase catalyzes the breakdown of starch into sugars in the mouth and the small intestines. Proteases catalyze the breakdown of proteins into amino acids in the stomach and the small intestines. Lipases break down or catalyze the breakdown of fats and oils into fatty acids and glycerol and are just found in the small intestines. Over time, we've found more and more uses of enzymes. At this point, it's worth noting down that anything that you want to break down with an enzyme, you simply take the name of it and put A's at the end. So if you want to break down lipids, you'd call it lipase. To break down proteins, you'd call it protease. To break down carbohydrates, you'd call it carbohydrase. If you're breaking down maltose, it will be called maltase. If you're breaking down fructose, it will be called fructase. It always follows the same pattern. Enzymes allow certain industrial processes to be carried out at normal temperatures and pressure, thereby reducing the amount of energy, so therefore the expense needed to carry out the reaction. Enzymes can also be used in the home in biological detergents. So, proteases can be used to pre-digest enzymes and are used in the manufacture of baby foods. Lipases are used in biological washing detergents as fatty stains can be broken down by them. Carbohydrates can be used to convert starch syrup, which is relatively cheap, into sugar syrup, which is more valuable. And isomerases can be used to convert glucose syrup into fructose syrup. Fructose is sweeter than glucose, so therefore can be used in sweeteners and can make foods taste as sweet with a lower amount of glucose being present in it. I hope that you found this video helpful. Remember, when revising, start earlier than later. Make sure that you are asking yourself questions, use flashcards, and quality of revision is better than quantity of revision. Look out for the other videos on additional biology, as they will help with other sections. Good luck with your exams.